I'm right, there's a huge amount of biology still to be discovered, processes well beyond what you'll find in the textbooks today. The experimental facts are already clear. Neo-Darwinism is dead.
All right, we're doing this. Folks, we need to talk about Denis Noble. If you're like me and you keep an eye on the portrayal of evolutionary theory in the popular press, you've likely come across Denis Noble several times in recent years. He was catapulted to the public limelight in 2022 after he had a debate with Richard Dawkins over the selfish gene at the How the Light Gets In festival. Since then, he's appeared on tons of podcasts and most recently was featured in a Forbes article titled "Evolution May Be Purposeful and It's Freaking Scientists Out." Noble has made a name for himself since his retirement in 2004 as a major critic of what he calls Neo-Darwinism.
No, the fact is that I think Neo-Darwinism is dead.
He, as well as the biochemist James Shapiro, have led the charge for the third way of evolution. On their website, they position the third way as sitting between Neo-Darwinism and intelligent design creationism. They write, quote, "The vast majority of people believe that there are only two alternative ways to explain the origins of biological diversity. One way is creationism, that depends upon intervention by a divine creator. That is clearly unscientific because it brings an arbitrary supernatural force into the evolution process. The commonly accepted alternative is Neo-Darwinism, which is clearly naturalistic science, but ignores much contemporary molecular evidence and invokes a set of unsupported assumptions about the accidental nature of hereditary variation. Neo-Darwinism ignores important, rapid evolutionary processes such as symbiogenesis, horizontal DNA transfer, action of mobile DNA, and epigenetic modifications. Moreover, some neo-Darwinists have elevated natural selection into a unique creative force that solves all the difficult evolutionary problems without a real empirical basis. Many scientists today see the need for a deeper and more complete exploration of all aspects of the evolutionary process." This framing is familiar. It echoes the sentiments of many of the extended evolutionary synthesis crowd that I critiqued in my video, "Is Evolution a Theory in Crisis?" Indeed, Noble frequently cites EES literature and even borrowed their Venn diagram in his 2015 article in the Journal of Experimental Biology. The only difference here is that Noble has added these colored words to the modern synthesis circle. DNA inheritance only, gene selection only, and genome isolated. The corresponding colors on the wider integrated synthesis are the supposed necessary extensions to account for the fact that these three things are now known to not be the only forms of inheritance and selection. So, standard evolutionary theory, which he likens to Neo-Darwinism, is missing stuff. This is a tune we're all too familiar with. None of the mechanisms listed in the wider diagram are missing from contemporary evolutionary theory, and in fact, most of them were known before the discovery of DNA. The only exception is evo-devo, but even the basic principles of that field were investigated statistically in quantitative genetics. We just didn't know the molecular details. But ideas like developmental stochasticity were fundamental parts of how the early geneticists were thinking. Just check out this diagram from Sewell Wright in 1922. The lines here show the multifaceted nature of inheritance. You can see influences from the environment, development, and the stochastic nature of Mendelian inheritance. Again, while they didn't know the specific details, they were already thinking about these things in the 1920s. We won't dwell on these specific extensions as I've covered all of this in a previous video. Instead, we're going to focus on the ways in which the third way crowd is different from your typical EESer. Noble and his third way acolytes have four main critiques of modern evolutionary theory. First, that traits acquired during your lifetime can be passed on to your offspring, and that this process drives adaptive evolution. That is, that Jean-Baptiste de Lamarck wasn't wrong about the inheritance of acquired characters. Second, they claim that organisms are purposeful agents of their own evolution, not the products of random mutation and natural selection. Related, thirdly, that organisms purposefully modify their own genome to respond to the world around them, a process they call natural genetic engineering. And lastly, that there is no such thing as selfish genes, that we are not products of our genes, and therefore any theory of evolution that is gene-centered is faulty. In addition to these four central claims of the third way, there's a great deal of historical revisionism that takes place in virtually all third way writings that creates an extremely skewed view of the development of evolutionary theory. If we take their historical presentation at face value, it reads like, people in the past were dumb, new discoveries overturned their dumb ideas, but modern people are too ideologically biased to accept new discoveries. Most evolutionary biologists have never heard of the third way, or even know who Denis Noble is, so I have mostly ignored him and his colleagues as just like contrarian cranks, and have focused my attention on the more reputable EES philosophers. But I've begun to notice a disturbing trend. While Noble has made exactly zero progress swaying evolutionary biologists, he seems to be making headway with the general public. Many of the videos and podcasts he's featured in have gotten hundreds of thousands of views, some with thousands of comments beneath, most of which are positive and praise Noble for speaking out. I fear the amount of media attention he's gotten has created a lopsided view of his reputation within evolutionary biology. Since the media is content with just letting Noble slander evolutionary theory unchecked, I fear it has fallen to me to try and deliver some balance to the discussion. This feels increasingly pressing. For example, the creationist think tank, the Discovery Institute, in their blog Evolution News, has written 3 articles about him this year alone, all praising him for standing up to the evolutionary establishment. Evolutionary theory, as I've long contended on this channel, is an extremely robust and mature science. To claim that it is dead or in need of radical revision is an extraordinary claim. Let's see if Noble has extraordinary evidence. Language changes through time. Words don't have fixed meanings, and there's really nothing to be gained in wading through the swamp of a semantic debate. Critics of the modern synthesis often call it Neo-Darwinism, a trend that goes back to Conrad Waddington in the 1950s. The paleontologist Stephen Jay Gould did the same thing, and ultimately many of the supporters of the modern synthesis, including yours truly, have adopted the mantle and used the words interchangeably. But for this discussion, it is helpful to distinguish them, at least historically, because Neo-Darwinism preceded the modern synthesis by over 50 years, and it lays the groundwork for much of Noble's critique. In the 1860s and 70s, in the immediate decades after Darwin's Origin of Species was published, there were heated debates over the mechanism of inheritance. This was because natural selection is not substrate independent, despite what Dan Dennett would have you believe. It only works under certain kinds of inheritance. At the time, blending inheritance was in vogue, and this created a major issue for Darwin's theory. The issue was first pointed out by Fleming Jenkins in 1867 in a review that he wrote of Origins. Jenkins was critiquing a particular example that Darwin gave in Origins of a population of wolves in which a brand new variant emerged and was favored by natural selection. This hypothetical served to contrast the case in which the environment changed and existing variation suddenly became favorable. Jenkins notes that under blending inheritance, this single individual with a brand new favorable trait would have to mate with a wolf that didn't have the trait, and so their offspring would only have half the benefit. Since they could only have half the benefit, selection on them would be half as strong, and subsequent matings would wash out the trait further until future selection was overpowered by inheritance. This observation by Jenkins startled Darwin so much that he removed the example from later editions of Origins. Darwin had a big inheritance problem. If he couldn't come up with a theory of inheritance that could overcome the effects of blending, natural selection might be dead in the water. What Darwin would settle on and flesh out in his 1868 book, The Variation of Animals and Plants Under Domestication, is known as the theory of pangenesis. He argued that organisms are shaped by their environment. If you spend a lot of time in the sun, you get more tan. If you work all day at an anvil, you get bigger biceps. And that when it comes time for reproduction, the cells of the body, called the soma, produce microscopic gemmules. These gemmules carry with them information from the soma to the germ cells and directly modify them with this new information, allowing organisms to pass their environmental modifications on to the next generation. Now, he still thought there was blending inheritance between parents, but what pangenesis did was restore variation each generation. If the environment was consistently applying the same pressure, then the variation would persist and selection could keep favoring it. In this way, the emergence of the variation and the favoring of it by selection were the same process. Both were directed by the environment itself. This is key to understanding why Darwin and those who would eventually become known as the Darwinists believe that natural selection was a creative force. As an external environmental pressure, it both acted to supply the variation as well as mold it into the future. The idea that traits are shaped by the environment directly is known as the inheritance of acquired characters and was popularized by Jean-Baptiste de Lamarck in 1809, and indeed this mode of inheritance is often called Lamarckian inheritance. From this, we see that Darwin was quite the pluralist. He believed that natural selection was the primary force of evolution, but he readily accepted other forms as well, and his theory of inheritance was, in effect, a mechanistic hypothesis of Lamarck's idea. While Lamarckian inheritance was popular among naturalists, indeed, even into the 1940s, a lot of naturalists favored it so much that Ernst Mayr had to dedicate a ton of space in his book Systematics and the Origin of Species to explaining why they were wrong. There were two key developments that undermined it. The first came from Darwin's own half-cousin, Francis Galton. He thought that if Darwin was correct about these gemmules, then he should be able to change the traits of organisms by swapping their gemmules. To him, it was conceivable that these were carried in the blood, since that was the fluid medium that transports hormones and oxygen around the body, and thus was likely to also transport these gemmules. He thus transfused the blood between two breeds of rabbit and then examined the traits of their offspring. To him, if the gemmules carried the information of inheritance, then the offspring shouldn't look at all like their parents, but like those of the donor blood. Of course, he didn't see this. The offspring were just like their parents. The second attack on pangenesis came from the German biologist August Weismann. Weismann is responsible for germ plasm theory, which is often credited for the concept of the so-called Weismann barrier. This is the idea that only the germ cells carry the information of inheritance and that they are uninfluenced by the soma. There is a barrier between them. He tested his idea, and by extension Darwin's theory of pangenesis, by performing a rather morbid experiment in which he cut off the tails of dozens of mice and had them breed. He continued this over multiple generations, but never did the tails of the mice get any shorter. That is, the environmental pressure of losing a tail was never transmitted from the soma to the germline. Thus, Darwin's theory of pangenesis rapidly fell out of favor, but it wouldn't be replaced by a new theory of inheritance until the rediscovery of Mendel in the early 1900s. And this rediscovery was seen as vindication of Weismann's ideas. Changes in the heritable material was internal to the organism, that is, caused by mutations directly to the germline, not caused by the environment, which was external to the organism. This distinction of internally directed evolution was championed by the self-styled mutationists like Hugo de Vries, Thomas Hunt Morgan, Reginald Punnett, and William Bateson, all giants in developing the theory of Mendelian inheritance. The term Neo-Darwinism was coined by George Romanes to serve as a distinction between Darwin's concept of natural selection and that favored by Weismann. I should note here that Alfred Russel Wallace, the co-founder of the theory of evolution by natural selection, also fell into the Weismann school of thinking. To Romanes, Darwinism referred to the pluralistic concept of natural selection acting on environmentally acquired characters, as well as other concepts like Lamarckian use and disuse, whereas Neo-Darwinism was the more restricted idea of Weismann and Wallace that natural selection acted on variation that emerged in the germline, protected from the soma. Thus, variation emerged internally and was then selected externally. This separated selection from the process of creating variation, something the mutationists would latch onto in their later writings. Now, when Noble claims that Neo-Darwinism is dead, one thing he is in fact referring to is the Weismann barrier. A large part of his critique is that the soma does direct variation in the germline. Indeed, he has called for a return of Lamarckian-style inheritance, something long dismissed by the modern synthesis. Why did they dismiss Lamarckian inheritance? The answer to this is often omitted from Noble's writings and other supporters of Lamarckian-style inheritance, so it's worth engaging briefly here. We'll expand on it and more modern work in the preceding sections. A great place to start is with the Danish biologist Wilhelm Johansen. He is responsible for the terms genotype and phenotype, and his most famous experiment demonstrated the inability of populations to adapt in the absence of genetic variation. He worked on self-fertilizing beans and was able to remove all genetic variation by having them only reproduce with themselves for many generations. Importantly, while they lacked any genetic variation, they still had phenotypic variation that was roughly normally distributed. What he observed was that when there's no underlying genetic variation, as in the case of pure lines, even the most extreme phenotypes every generation would regress to the mean. That is, they would consistently become more and more like the average value, demonstrating that the population was incapable of changing. This led to an extremely important addition to the concept of evolution. The variation between individuals must be heritable. Second, it demonstrated that consistent environmental pressure was not enough to permit evolution to occur if there was no heritable variation. If Lamarckian-style inheritance was important, these beings should have been able to evolve by acquiring traits from their environments. But they didn't. Without genetic variation, that is, variation in DNA as we understand it today, populations fail to adapt further. This was also established by many farmers and livestock breeders in the late 1890s and early 1900s. Thomas Hunt Morgan, who won the Nobel Prize for the discovery of the chromosomal basis of heredity, noted the importance of mutations to resupply variation to populations. He observed many farmers rapidly depleted the genetic variation in their crops, which initially led to a rapid increase in yield, but that it would plateau, and then no further improvement was possible once the genetic variation was depleted. Further improvement, he noted, would have to wait on future mutations. Yet again, if Lamarckian inheritance were the dominant cause of trait variation, these crops, under consistent environmental pressure, should still be able to adapt without genetic variation. So it was well known to the architects of the modern synthesis that adaptation was limited to genetic variation. A few lab experiments helped to demonstrate that genetic mutations are random with respect to the need of the organism, not induced by the environmental pressure itself in the direction of the need. The first was the Luria-Delbruck fluctuation test in 1943. They directly tested the Lamarckian versus the Darwinian explanation for adaptation by testing bacterial resistance to a T1 phage virus. They reasoned that if resistance was induced by exposure, then each agar plate of the bacteria should have roughly the same number of resistant bacteria at the end of the experiment. If, however, resistance arises from a mutation that is random with respect to the need of the bacteria to evolve resistance, then there should be a high variance across replicas regarding how many bacteria will have the mutation. What they found was a very high variance across replica plates for the acquisition of resistance, suggesting that random mutation followed by selection was the mode of adaptation, not Lamarckian inheritance. A similar experiment was performed by the Lederbergs in 1952. They smeared agar plates with bacteria and allowed the colonies to grow. Then they made copies of each agar plate using a technique called replica plating, in which you stamp the agar plate and transfer part of the colonies to a new plate. This allows you to, in effect, replicate the colonies of the original plate. After doing this, they dosed the replica plates with penicillin. Most of the colonies in the plates died, but a few had resistance and survived. The question is, did these colonies evolve resistance in response to penicillin, as Lamarckian inheritance suggests, or did they already have the variation ancestrally, as the Darwinian explanation predicts? To find out, they dosed the original plates with penicillin as well. If the traits were acquired in response to penicillin, then the colonies that evolve resistance shouldn't necessarily be the same ones from the replica plate that survived. You would expect them to be random. But in the case of standing variation, the exact same colonies that were resistant to penicillin in the replica plate should be resistant in the original plate. And that is exactly what the Lederbergs found. Thus, the rejection of Lamarckian inheritance by the modern synthesizers is predicted on two key observations. One, the failure of selective breeding experiments in the absence of genetic variation, and two, laboratory experiments demonstrating that selective pressure doesn't induce mutations. To review, Neo-Darwinism is the perspective, supported by Weismann and later proponents of the modern synthesis, that the germline is distinct from the soma, and that changes to the germline happen internal to the organism, not acquired from the environment. Thus, natural selection acts upon mutations to the germline that are random with respect to the organism's need for them. Now that we've established a bit of the historical background, we should understand at the most basic level what Noble is calling dead when he claims that the modern synthesis or Neo-Darwinism is dead. So what exactly has died? What are the fundamental failings of these ideas? The big one to Noble is the Weismann barrier. He has called for a complete reversion in our thoughts about Lamarck, a triumphant return of that maligned French naturalist. He appeals to epigenetics as the new molecular mechanism for the acquisition of environmentally induced adaptive traits. But what exactly is epigenetics? It's not as clear-cut as you might think, and different people have used the term differently throughout history. This has led to considerable confusion in the literature. So first, let's examine the different ways in which the term epigenetics has been employed. To do so, we should return to one of the first people to popularize the term in the 1940s, a man we've already mentioned in passing, Conrad Waddington. In 1942, the year Julian Huxley published Evolution, the Modern Synthesis, which provided the new paradigm its name, the British embryologist Conrad Waddington sent a short letter to the journal Nature titled Canalization of Development and the Inheritance of Acquired Characters. He began the paper with the following, quote, "The battle which raged for so long between the theories of evolution supported by geneticists on one hand and by naturalists on the other has in recent years gone strongly in favor of the former. Few biologists now doubt that genetical investigation has revealed, at any rate, the most important categories of hereditary variation, and the classical naturalist theory, the inheritance of acquired characters, has been very generally relegated to the background because the existence of which there was no adequate evidence." He would go on to propose a mechanism by which he believed environmentally induced characters could be solidified in populations. He called this mechanism genetic assimilation. His argument went like this. The early geneticists often referred to common forms in nature as wild types, and those not matching them as mutants. Waddington noted that despite the occasional appearance of these mutants, they were remarkably rare. Indeed, populations could be exposed to variations in the environment and possess lots of genetic variation, but still develop as the so-called wild type or common form. To Waddington, this meant two key things. First is what he called canalization. This is that despite many interacting genes and variable environments, development funnels organisms into the same phenotype. In effect, developmental pathways are extremely robust to perturbations from both the environment and genetic mutation. R.A. Fisher's theory of the evolution of dominance provided one explanation for this, in which selection has favored genes that can modify the degree of dominance in interacting genes, cranking up or tuning down expression depending on the genetic or environmental background. Others, like Herman Muller, pointed out that in sex-linked genes, there was a mechanism that is called dosage compensation, in which females with two X's don't make twice the gene product of males. Instead, one of the X's is silenced. Furthermore, we know today that loss-of-function mutations can be compensated for by possessing many copies of the same gene, which makes gene regulatory networks both highly redundant but also robust. All of this led Waddington to suggest that most of the action of natural selection was on gene regulatory networks. Since many mutations existed all the time, selection only really sees the sum of all their genetic effects and acts to tune them at the regulatory level. In this way, selection favors canalization. Furthermore, this means that there could be a lot of genetic potential for relatively large phenotypic shifts in a population kept at bay by this canalization process. He envisioned development as a marble rolling down a hill. Where the marble ends up is the final phenotype, and it is directed by the shape of the hill along the path to the lowest point. The shape of the hill is the combination of environmental and genetic effects, including regulation. Selection ultimately shapes the stability of the final basin of attraction, but he imagined the case in which environmental shifts might change the topography of this developmental hill, which, depending on how early this happens, could dramatically alter where the marble ends up. Importantly, this is not a fitness landscape where the hillside is some kind of unrealized potential that must wait on mutation to be explored. Rather, it is what already exists genetically, only that the current timing of switches and extent of regulation ensure the normal outcome, which is taken to be the most common basin. Thus, dramatic shifts in the environment alter where the marble ends up without changing the underlying genotype. If the shift is favorable to the organism, selection favors regulation that follows this path, which continues so long as the environmental pressure persists. Eventually, the environmentally determined pathway becomes genetically assimilated, when the underlying genetic variation shifts such that this pathway becomes the new normal development. Thus, genetic assimilation is the idea that plasticity precedes genetics. Traits that can be shaped by the environment first change in response to environmental pressure, and then only later become cemented by either shifts in regulation or novel genetic mutations. Waddington thus defined epigenetics as the connection between genotypes and phenotypes. In particular, it is the interaction between DNA sequences and other cellular components to regulate and ultimately give rise to phenotypes. In this way, Waddington saw epigenetics as akin to development, and that the inheritance of environmentally induced traits occurred by genetic assimilation. If we define epigenetics as Waddington did, then any kind of gene regulation counts as epigenetic modification. The first molecular basis of gene regulation was worked out by French biologists Francois Jacob and Jacques Monod, and in 1965 they would win the Nobel Prize in Physiology for their work. They managed to decipher the regulation and expression of the lac operon, a collection of genes in E. coli that are responsible for signaling the transport and metabolism of lactose. In the preceding decade, they discovered that the lac operon is composed of a promoter, an operator, 3 genes known as lac-Z, lac-Y, and lac-A, and a terminator. Upstream of this operon is a gene that codes for a repressor of the operon, lac-I. When this is active, it binds to the promoter and prevents expression of the operon. This is a kind of epigenetic modification in Waddington's definition. What they found was that the trait, metabolism of lactose, depended on whether or not the operon was epigenetically modified. That is, there was no change in the DNA sequence, but sometimes the bacterium expressed lactase and sometimes it didn't, depending on its epigenetic state. This epigenetic state was a direct response to the environmental presence of lactose. You might notice some similarities between Waddington's ideas and the concept of phenotypic plasticity. That is, the ability of an organism's phenotype to respond plastically to different environmental pressures. In the case of E. coli, the lac operon allows it to produce lactase for the digestion of lactose, but it only does so in the presence of lactose. Now, the astute observer has probably noticed that none of this really has anything to do with Lamarckian style inheritance. The environment has not induced a heritable trait in Waddington's model. Rather, the traits themselves are plastic, respond to the environment, but must wait on genetics to eventually cement them in place. In 1958, over a decade after Waddington coined the term epigenetics, David Ledbetter Nanny expanded Waddington's original definition to include modifications of gene expression states across cell cycles. That is, a mitotically dividing cell would relay its epigenetic state to its daughter cell. Hence, epigenetic modification was not relegated to a single cell. The gene didn't need to be remodified after every cell division. Nanny contended that there was a kind of stability of gene modulation because cells would pass on their epigenetic states. Work by folks like Robin Holliday in the 1980s on cellular memory, gene expression, and DNA methylation all indicated that these states could persist across mitotic events, supporting Nanny's expanded definition. Indeed, in 1994, Holliday provided the definition of epigenetics that is closest to the one that is presently debated with respect to the possibility and importance of Lamarckian-style inheritance. He defined it as, quote, nuclear inheritance, which is not based on differences in DNA sequence. This is a bit too general to be very useful. A clearer definition is provided by Wu and Morris in 2001, quote, the study of changes in gene function that are mitotically and or meiotically heritable and that do not entail change in DNA sequence. Now, we see a fundamental shift from Waddington's original usage to those by Nanny and later by Holliday. The shift is inheritance. Waddington was only concerned with how genotypes interact with other cellular components to give rise to phenotypes developmentally. Now, epigenetics had become the study of the inheritance of these modifying cellular components that are held to be independent of nuclear DNA. For the remainder of our discussion about epigenetics, we're going to follow Holliday's 1994 definition, because this is the one most closely linked to the concept of Lamarckian inheritance that Noble is trying to resurrect. As pointed out by Carrie Deans and Keith Maggart in their article, What Do You Mean, Epigenetics?, published in Genetics in 2015, this shift in definition kicked off an entire new field of investigation by which all weird phenomena that couldn't easily be attached to a genotype would fall. This included things like imprinting and maternal effects, in which the phenotype of the offspring depended not on the genotype, but on the phenotype of the mother, as well as things like position effect variegation, which is the change in the expression of a gene depending on where that gene falls on the chromosome. As a personal anecdote, when I started graduate school in 2016, the word epigenetics was everywhere. Anytime you couldn't explain something as a clear result of a gene, you'd say, well, it's probably epigenetics. It became a kind of scapegoat, a way to sidestep a difficult problem. Oh, your genome-wide association study didn't find any hits? It's probably because the trait is epigenetically controlled. Tons of folks in my cohort were interested in studying epigenetics, and many people were talking about it as if it was some new mechanism of evolution. This leads us to a key question about the basic role that epigenetics has played in the development of evolutionary theory. To understand this, we need to remember that evolution, while intimately connected with heredity, is a theory about the forces of change of populations over generations. This can technically occur in both Mendelian and non-Mendelian systems of heredity, and there exists a breadth of mathematical tools at our disposal for evaluating each. In the next two sections, we'll address the two core features of Noble's first critique of Neo-Darwinism, his call for a return of Lamarckian inheritance. The first feature is that Neo-Darwinism, which I'm going to synonymize with contemporary evolutionary theory, is both ill-equipped to deal with epigenetic inheritance and that it has been ignored by evolutionary theorizers. His second claim, which is really the only one of his claims that would actually necessitate rethinking of the modern synthetic paradigm, is that the environment doesn't just influence epigenetic modifications, which we all agree that it does, but that these inductions are themselves adaptive. Now again, for this to be a challenge to the modern synthesis, the propensity to respond adaptively to the environment cannot be genetically programmed. That is, natural selection can shape gene regulatory networks such that they can themselves be responsive. If this is the case, then we're just dealing with plasticity, not Lamarckian evolution. Lamarckian evolution requires that the environment induced something fundamentally new, not merely expressed a plastic response that selection already cemented in place. Before unpacking this further, let's situate epigenetics within evolutionary theory as it is currently understood. Then we'll be in a position to deal with Noble's Lamarckian ideas head-on, as we will have distinguished them from what is already accepted within the standard paradigm. In 1956, the economist Kenneth Boulding wrote, "Somewhere between the specific that has no meaning and the general that has no content, there must be, for each purpose and at each level of abstraction, an optimum degree of generality." Evolutionary biology has grown to become a monstrous discipline, so big and so encompassing that many individual biologists can be just buried under all the specific details of their subfield. R.A. Fisher lamented this fact in 1915 when he wrote, quote, "The tendency of modern scientific teaching is to present masses of detail of doubtful truth and questionable weight in such a way as to obscure principles." Biologists, especially those like Denis Noble, tend to embrace the specifics. We love to dive into the nitty-gritty details of whatever system at whatever level of biological hierarchy we're studying. So much so that we think that whatever we're looking at this moment in time is somehow of paramount importance to understanding the evolutionary process writ large. But the fact of the matter is that at the most fundamental level, evolution is a theory of change in the heritable characteristics of a population. All the rest is details. Thus, if we want to situate epigenetics, or more generally non-genetic inheritance in evolutionary theory, we should start from first principles. If you're a regular viewer of this channel, you'll know that I consider the price equation as the fundamental theorem of evolution. It begins with a representation of a biological system, ancestors, and their descendants. These ancestors have some trait value that they then pass on to their descendants, with a potential for some level of deviation between parents and offspring. From this basic model, which we believe represents any kind of biological system you can imagine, we can derive a series of identities. For the full derivation, see my video on the math of Fisher's fundamental theorem, linked in the description, and if you'd like a history of the price equation, see my video, Evolution's Fundamental Theorem. What we end up with is an equation that looks like this. Delta Z bar, which is the change in the mean trait of the population, is equal to the covariance between the fitness of the ancestors, which is simply how many descendants they have, and their exact trait value, plus the average difference between ancestors and descendants, all divided by the mean population fitness. The first term, the covariance between ancestral fitness and the trait value, is both natural selection and genetic drift. We can partition this term into two components to see why. The first component, covariance, is when fitness is causally related to the trait, such as having a larger beak allows you to crack bigger nuts and get more nutrients, and that allows you to have more children. This is, of course, just natural selection. The other is when fitness and a trait are randomly associated. For example, someone with brown hair gets struck by lightning. The number of offspring they have is now reduced, and hence the potential for people to be born with brown hair in the next generation has gone down. But it's not because brown hair gave them a survival disadvantage. Fitness and the trait are just randomly associated. The second term is the deviation between parents and offspring with respect to their trait value. This represents several biological phenomena, including mutation, recombination, but also selection at lower levels. Now, you'll notice that we haven't said anything about the specific mode of inheritance. That's because the Price theorem makes no assumption about whether inheritance is genetic, epigenetic, cultural, whatever. All you need is a covariance between parents and offspring. Furthermore, it says nothing about what the trait itself is. And this latter fact will allow us to expand the price theorem to consider both genetic and non-genetic inheritance. So let's build a price equation style model that includes genetic and non-genetic effects. Furthermore, we can simplify it a little bit and consider the relationships as direct parent and offspring instead of ancestor and descendant, the latter of which is agnostic to the time interval between the two. And this way we'll also want to model both the probability that the parent survives into the next generation in addition to how many surviving children they have. Thus, first off, we should note that the total number of individuals in the population is W equals to B plus P, where B is the product of the number of offspring produced and the probability they survive, and P is the probability that the parent survives. Next, we note that the offspring may differ from their parents, and how they differ might depend on whether those differences occur genetically, which will denote G, or epigenetically, which will denote H. These are, in effect, the trait values of the parent, so children are G plus G delta B, where G delta B is the expected genetic differences between parent and offspring, and H plus H delta B, where H delta B is the epigenetic differences. Importantly, since we've allowed parents to live across generations, they can change in their genetic or epigenetic state over their lifetime, and these changes would then be heritable to future offspring. We'll use delta GP and delta HP to denote the expected difference in G and H within parents after versus before the parent survives to the next generation. Now, I'm going to show you the steps of how we can derive a price equation-like result from these basic definitions. These derivations were originally done by Troy Day and Russell Bondaryansky and the American Naturalist in 2011. Notice how equations 1a and 1b are effectively the original price equation, but partitioned by genetic and non-genetic inheritance. The first term is still taken as the covariance between fitness and, in this case, the genes of an individual. By genes of an individual, we should understand that to mean their phenotype as expressed by the underlying genotype. We then have two terms, the average genetic difference between parents and offspring in the present generation, what we call reproductive transmission, and the mean genetic difference from parents surviving longer, what we might call survival transmission. This allows for the fact that epigenetic states are modified during one's lifetime and might then be passed on to future generations. Though notice the same relationship is seen for normal genetic states as well. This accounts for things like age effects of parents. We thus see the same identities between genetic and non-genetic inheritance. This reiterates an important feature of the price equation. It doesn't matter what the mode of inheritance is. They're all modeled exactly the same way. But even more importantly, it shows us that the mechanisms of evolution that can be elucidated from the price equation are fundamentally the same. The covariance term, which represents the removal of variation by drift and selection, is the same. But so are the change in the transmission terms. Both genetic and non-genetic inheritance change in the same way and by the same mechanism. That is, mutation. Prior to the discovery of DNA, mutation was defined as the change in the heritable material, which they just called a gene. Modern biochemistry has since narrowed that definition to only refer to change in the DNA sequence, but in our version of the price equation, we see that mutations, again just heritable changes, extend to non-genetic characters as well. Let's be clear on the implications. What this derivation of the price equation, which includes non-genetic inheritance, demonstrates to us is that non-genetic inheritance, which includes epigenetics but also cultural inheritance, is not a novel kind of evolutionary mechanism. It is instead, as the name implies, a novel kind of transmission or inheritance. But the same evolutionary processes that are explicable from the price equation are at play. That is, selection, drift, and mutation. The first two cull variation, and the latter replenishes it. But there are other really cool things we can learn here. First, we've completely partitioned these terms into separate modes of inheritance, but obviously they interact with each other. So we really need an additional covariance term that captures the covariance between genetic and non-genetic inheritance. The following derivations show the change in mean genotype, which is delta G bar, and mean non-genetic type, delta H bar. The sigma term is now either a variance or a covariance, depending on the subscript. subscript, and the beta term is the selective gradient acting on either G or H, depending on the subscript. Thus, the first term in equation 2a is the variance in genes, sigma GG, multiplied by selection acting on genes, beta G. The second term is the covariance between genes and non-genetic effects, sigma GH, multiplied by the indirect selection acting on genes. This effect only occurs if there's a non-zero covariance between genetics and epigenetics. Otherwise, sigma GH is 0 and this term drops out of the equation. A few fascinating things come out of this. First, as the price equation has always excellently shown, evolutionary dynamics of populations are dependent on the interaction between modes of transmission and selection. Since the selection gradient is a component of the mean of the genetic and non-genetic variables, a change in non-genetic factors can alter the adaptive landscape of genetic factors and vice versa. Furthermore, we see that there can be transmission interactions between genetic and non-genetic factors influencing the parent-offspring deviations, directly modifying the probability of getting a mutation. We even know of biological processes that directly affect the mutation rate in this way. A classic example is methylation of CpG sites. Methylation is a type of epigenetic modification of a cysteine guanine site, but when this occurs, it increases the probability of a mismatch mutation, because 5-methylcytosine readily deaminates to thymine, which is often repaired to AT instead of the original CG. Thus, epigenetic modification can increase the mutation rate of genes, a biological process that is captured in the other terms of equation 2a and 2b. Day and Bondariansky conclude, quote, "Clearly, the diversity of inheritance mechanisms, that is, proximate pathways mediating the effects of parents on offspring, is very large. However, these mechanisms engender a much more limited array of inheritance patterns, that is, unique combinations of reproductive transmission and survival transmission rules. The theoretical framework provided by equations 1 can therefore be used to investigate the evolutionary consequences of a great diversity of inheritance mechanisms, regardless of the proximate details of transmission." The "regardless of the proximate details of transmission" is the critical phrase here. Whether changes are being caused by genome duplication, transversion, polymerase slippage, gene conversion, segregation distortion, methylation, whatever, the core theoretical framework is fundamentally unchanged. The proximate details don't matter. A mutation is a mutation. Transmission is transmission. Evolution is the change in the heritable material in a population across generations, whatever that heritable material might be, whether it's restricted to the gene or to other cellular components that modify that gene. J.B.S. Haldane made the same point in his famous 1964 paper titled A Defense of Beanbag Genetics. He, along with Sewell Wright and R.A. Fisher, were being lambasted by Ernst Mayr for not considering the physiological interaction between genes in their mathematics. He pointed out that if they also incorporated every single biochemical detail, that is, every proximate mechanism, then the field would cease to be evolutionary biology. It would just be biology. Thus, non-genetic inheritance is not a forgotten mechanism of evolution. In his review of Kevin Lala and friends' recent book, Evolution Evolving, in which they critique the modern synthesis and argue that things like developmental bias is a missing mechanism of evolution, David Hull provides us a clear roadmap for when we should think of something as an evolutionary principle. He notes that evolution in any form must start with some system of transmission, irrespective of what that is. Then you need something that causes variation among replicators of that system, i.e. mutation. Once there's variation, you can have selection and drift. He writes, quote, "This suggests a clear criterion for when a phenomenon or process should be elevated to a principle. If it is a key component of evolutionary thinking, but does not have a likely evolutionary explanation, we should treat it as a principle. Systems of inheritance are a prerequisite for evolution, but they are not themselves causes of it." Hopefully you've seen thus far that epigenetic transmission can be very easily incorporated into modern evolutionary theory, and indeed already has been. I've provided a more modern example, but various other kinds of non-genetic inheritance have been discussed for a long time in theoretical population genetics. Sewell Wright dedicated several chapters to non-genetic inheritance in his famous 4-volume work from the 1970s on genetics and evolution, including detailed derivations for modeling maternal effects and cytonuclear incompatibilities. Nothing we've discussed thus far is outside of the modern synthesis, or at least outside of contemporary evolutionary theory. Indeed, in the version of the price equation we just derived, adaptation occurs solely through natural selection, and hence it still rejects a Lamarckian mode of evolution that Noble favors. Now that we've situated non-genetic inheritance within the framework of evolutionary theory, let's get down to some specifics. Demonstrating that we can model it is not the same thing as showing it even exists. While we were setting aside sort of the proximate details for the moment while deriving equations, when it comes to biology, we do still need to show that there is some biological basis for these ideas. So what is the current state of study within evolutionary biology with respect to transgenerational epigenetic inheritance? And do we have evidence that it is consistently biased in favor of adaptive phenotypic changes? That is, where does biology stand today with respect to Lamarckian evolution? Three giants in population genetics, Deborah and Brian Charlesworth, along with Nick Barton, published a review of the subject of what they call Neo-Lamarckism in 2017 in a paper titled The Sources of Adaptive Variation. In it, they note that for a radical revision of the modern synthetic paradigm to occur, Noble and the supporters of the Lamarckian view of evolution need to establish three things. First, quote, a given environmental treatment tends systematically to induce heritable adaptive epiallelic variants. That is, it needs to be shown that these epialleles are not dependent upon genetic alleles. It is often the case that there is both a change in epigenetic state that is causally connected to a change at the sequence level. And ruling this out is paramount to making the case for Lamarckian style evolution. Second, that natural selection is not involved in the spread of such variants through populations. If the environment induces an epigenetic change that is adaptive, but it's selection that causes it to spread, then all we've done is recreated the scenarios modeled by the price equation in the previous section. Lamarckian evolution supposes that environmental induction is itself a cause of adaptation, not merely an extra source of variation for Darwinian selection to act upon. And thirdly, quote, the variants in question can be stably transmitted for many generations in the absence of the treatment. We already know that epigenetic modifications can be inherited between mitotic divisions during development, and we have ample evidence that various kinds of epigenetic factors can be inherited meiotically as well, one of the earliest being maternal effects. But if this mode of evolution is going to rise to the same level of importance as genetic change, it needs to be at least as stably transmitted as genes are. If epialleles get washed out after a generation or two, they can't persist and impact long-term evolution. Let's start with our first point. We know that epigenetic modifications are key to developmental processes virtually exactly how Waddington visualized them in the 1940s. Alterations to epigenetic states effectively skew the shape of the developmental hill, modifying the final phenotypic expression. But as Waddington suggested, these epigenetic states are expressions of underlying genetic states. Indeed, his view of epigenetics was that they bridged the genotype to the phenotype. One of the most famous genes for controlling epigenetic modifications is DNA methyltransferase, of which there may exist several copies throughout the genome. But the gene itself is highly conserved across organisms. Most studies that have knocked out or modified the gene using CRISPR technology have found that methylation patterns are significantly altered. When the gene is removed entirely, methylation vanishes from the genome and often results in death. Some organisms, like Arabidopsis, are exceptions in that a knockout to one of the DNA methyltransferase genes appears to be compensated by others, reducing methylation but otherwise having no negative phenotypic effects. This indicates that the propensity for a cell to modify its epigenetic state is actually genetically controlled. This is exactly what we'd expect if developmental plasticity had been fine-tuned by natural selection. If having the capacity to respond plastically to environmental perturbations is beneficial, as it almost certainly is, then having a gene or genes that control expression patterns in response to environmental stimuli would be highly favored by selection. Thus, the ability to express, silence, or repress gene expression is not a function of an environmentally acquired trait in a Lamarckian fashion. These are all genetically controlled. As in the case of DNA methyltransferase or the famous lac operon, it is simply that these genes are capable of responding to the environment, and their ability to do so has been fine-tuned by natural selection. In a large review of the literature on epigenetics, Yi in 2024 noted that a growing body of work indicates that epigenetic alleles are tightly linked to genetic alleles. They go on to write, quote, "Therefore, any study of epigenetic variation between individuals or populations should take into account the underlying genetic variation as a source of the epigenetic variation." This is of course quite difficult to do, and most organisms don't have the genetic resources available to easily connect patterns of methylation with DNA sequences. But that in and of itself is a testament to how premature Noble's call for replacing the modern synthesis is. For most organisms, we simply don't have the necessary data to draw any broad conclusions about the connection between DNA variants and epigenetic variants. And for those that we do have this data, it is becoming increasingly clear that the latter relies on the former. Now, when these epialleles arise, ignoring whether they are connected to DNA variation, what is their functional significance? Are they systematically biased towards being beneficial? First, we've known since before the discovery of DNA that most mutations are harmful. And given the much higher rate of mutations in epialleles compared to genetic ones, it's likely that they both have a much smaller effect than genetic mutations and are likely mostly neutral or weakly deleterious. Studies in Arabidopsis, one of the few systems in which this has even been measured, found very weak selective effects on epigenetic mutations. Furthermore, in a large review of almost 500 studies where phenotypic adaptation was measured, Martin and Orgogozo in 2013 found only a single case of a potential epigenetic marker contributing to the adaptation. All the others were linked to DNA sequence variation. This echoes the old studies of the early 1900s that I mentioned previously. Farmers and livestock breeders have long known that if you remove genetic variation, your crop will cease to respond to selective pressure. This draws a strong link between adaptability and DNA sequence variation, and leaves quite the limited role for epigenetics in adaptation. For their second point, is there any evidence that natural selection is not responsible for the spread of the epiallele once it arises? The answer, unequivocally, is no. In all cases in which epigenetic systems have been elaborated, they have both a clear genetic underpinning and variations are favored or purged by natural selection. A famous example is the small interfering RNAs called piRNAs that act to protect the genome against transposon proliferation. The system is quite elaborate and fascinating and works like this. In the egg cell within the mother, prior to fertilization, piRNAs are produced from transposon sequences and they hover around in the cell, ensuring a kind of epigenetic inheritance, since the state of the mother's egg cell is what will dictate the future phenotype. Though note, of course, that piRNAs are still produced from DNA sequences, and thus rely on the underlying DNA variation. Each generation, these piRNAs are produced anew, responding to the clusters of transposons and then acting to silence them. In Drosophila, there's a new transposon called a P element, in which there is not yet an established memory in the females that lack this element. Thus, a mating between a male with the element and a female without it results in hybrid inviability, because the the maternal egg cell is not primed to produce the pi RNAs before fertilization, leading to the proliferation of the P element. Thus, females that are naive to the P element are disfavored when P is at high frequency, but are more fit when it's at low frequency. This is, of course, a case of frequency-dependent selection, and this entire amazing system is an example of DNA variation leading to epigenetic variation, which is then acted upon by natural selection. A classic tale of Neo-Darwinism. Lastly, how stably inherited are epigenetic markers anyway? Even if they are biased towards being adaptive and can be favored by natural selection, do they even stick around long enough to lead to sustained change in a population? Almost all the work on transgenerational epigenetic inheritance has come from the weed Arabidopsis and the nematode worm C. elegans. Johannes and friends in 2009 showed that epialleles were stably transmitted through 8 generations in Arabidopsis. The molecular mechanism of this is that germ cells in plants arise from somatic cells, and thus epigenetic inheritance might be more stable in plants than in animals. Indeed, the evidence for stable epigenetic inheritance in mammals is still inconclusive. Most studies on epigenetic inheritance in mammals, mostly in mice, have found that markers only persist for a few generations. In one study, they found that after 3 generations, the markers were still maintained, but only in some of the cells, indicating that they were gradually being lost. Thus, the present evidence for transgenerational epigenetic inheritance is mixed. It appears that in plants, it might be an important source of variation since the Weismann barrier is weak or non-existent in plants, given that their germ cells emerge directly from their somatic cells. But in animals, where the Weismann barrier is stronger, since germ cells are actually segregated from somatic cells, epigenetic markers are more likely to get reset each generation. Therefore, non-genetic inheritance is not a clear universal phenomenon, but varies across the tree of life. In addition, there's very little evidence that epigenetic variation is consistently decoupled from genetic variation, or that it consistently is adaptive when it is. Instead, the high rate of mutation for epialleles virtually ensures that the effects are likely very small and nearly neutral. Lastly, when epialleles are adaptive, their spread through the population is solely brought about by natural selection, just as predicted by standard evolutionary theory. Epigenetics is still a new and exciting field of inquiry. Future work will clarify the undoubtedly important roles of epigenetics in shaping trait variation and the ability of organisms to respond plastically to their environment. But no discovery thus far from anything in developmental or molecular biology has challenged the basic tenets of the modern synthesis or undermine natural selection as the sole force of adaptation. While the Weismann barrier has been shown to not be absolute, we've known that it wasn't since the first discovery of non-Mendelian patterns of inheritance, which have been known for over 80 years. None of this is to say that non-genetic inheritance isn't an important and fascinating field of study that future theoretical and empirical work should absolutely be dedicated to. What I'm offering here is caution against those who would claim that an entire paradigm is dead because of a handful of studies in Arabidopsis. Ultimately, good old genetic variation is still overwhelmingly the source of adaptive variation, and this has been verified by thousands of studies in both natural and laboratory populations. Therefore, Noble's attempt to resurrect Lamarck falls flat. This past summer in 2024, Andrea Morris, a journalist for Forbes, interviewed Denis Noble for an article titled "Evolution May Be Purposeful, and It's Freaking Scientists Out." In it, she writes, quote, "For Noble, this routine procedure offers clear evidence of the organism actively participating in its own evolution. It's doing natural selection. This is an alternative theory of evolution where cognition is fundamental. In this theory, the smallest unit of life, cells, have some version of intelligence and intent that allows them to detect and respond to their environment. He believes we're working ourselves into a sweat to exclude something so essential to evolution and to life as purpose and intention." The second critique of the third way crowd of standard evolutionary theory is that it ignores a few of their favorite buzzwords. These are purpose, agency, and teleonomy. That is, Noble and friends contend that organisms, including single-celled organisms, are purposive agents capable of directing their own evolution. They're goal-oriented and have the ability to make conscious or semi-conscious decisions about how to deal with the challenges that the environment throws at them. These ideas are either remarkably profound or extremely trivial, depending on how you define these terms. In a recent book, Evolution on Purpose, edited by Denis Noble and other third-way folks, Stuart Kauffman provides a definition of organismal agency as, quote, the capacity to define and regulate their own boundaries and sustain their integrity according to a set of internal motives and rules. He counts among agentive behavior the following: exploratory behaviors, migration to sources of nutrients or social cues, chemotaxis, cessation of movement, and quiescence. From this definition, we see that organismal agency includes everything from foraging to taking a nap. Whenever a bird decides to leave its roost and search for food, Noble and others would say that it is doing so on purpose and with intentionality. It's heading out with some foresight that wherever it's going, it's hoping to find food there. This is, of course, both true and extremely trivial. No one doubts that organisms behave in ways that can be defined as goal-oriented. In fact, the term teleonomy was coined by biologist Colin Pitterdrigg in 1958 to indicate apparent purposeness in organisms that is brought about by natural processes, including selection. Ernst Mayr adopted the term in 1965, but suggested the term be restricted to, quote, systems operating on the basis of a program of coded information. That is, goal-directedness in biology should be understood as emergent from underlying genetic mechanisms. Similarly, Richard Dawkins, in a talk titled Purpose of Purpose, used the term archaeopurpose, a purposive behavior evolved by natural selection. An example of this would be foraging for food, searching for mates, generally responding to external stimuli. While words like agency, purpose, and intentionality are terms too easily anthropomorphized for my taste, we can technically apply them to living things so long as we understand that we are using them in the apparent sense, not in the actual sense. That is, organisms behave as if on purpose. Not that the cells actually have some inherent purpose, since purpose implies a mind, and cells don't have minds. Again, all of this is trivial. What's it got to do with evolutionary theory, or more specifically with overturning it? As I mentioned before, the third-way supporters contend that evolution is itself purposeful, that organisms are shaping their own evolution on purpose. In a stunning example of historical revisionism, Noble has even gone so far as to claim that purposive evolution was originally part of Darwin's ideas, but was banned from evolutionary biology by people like Alfred Russel Wallace and August Weismann. In his 2021 article, The Illusions of the Modern Synthesis, Noble lists natural selection as one such illusion. But he means it in the most convoluted of ways. First, he notes that Darwin distinguished natural selection, the process of differential survival and reproduction, from sexual selection, which is an organism actively discerning between potential mates. He equates the latter form with artificial selection, which he calls intentional selection. In the case of artificial selection, humans are intentionally picking out traits they want. In sexual selection, individuals are intentionally choosing traits they want in a mate. He then makes the obvious point that nature doesn't select anything, so lacks intentionality, and that natural selection is an unfortunate metaphor. Of course, this has been pointed out by numerous biologists, including several architects of the modern synthesis. But he suggests that we have been genuinely fooled by the metaphor itself. He writes, quote, "It is an illusion with important consequences because it can lead people to think that real selection by organisms themselves with the power to do so cannot even exist." Not only is this false, it's a non sequitur. Natural selection is obviously non-intentional, but that fact doesn't mean that all forms of selection are. Furthermore, what we usually mean by selection isn't intentional is with respect to adaptation. Nature isn't trying to mold well-adaptive organisms, but neither is sexual selection. Mates aren't chosen because they're trying to build a more fit population. They often just find each other sexy. Indeed, this is exactly how Darwin meant it. He believed that organisms made mate choice decisions for purely aesthetic purposes, which might have no consequence to how well adapted to the environment they are. But Noble needs a boogeyman. There must be some figure who rejected Darwin's idea, and if so, he can attach all subsequent evolutionary biologists to it. He finds this character in Alfred Russel Wallace, who famously disagreed with Darwin's idea of sexual selection because he didn't believe that organisms would select mates without consequence to how well adapted they were. He then just equated natural and sexual selection because he believed both were leading to adaptation. The former was being done by the environment, whereas the latter was being driven by the organisms themselves and who they chose to mate with. Presumably, they were always picking the most fit. With this detractor in hand, Noble claims, quote, "If all evolutionary change produced by natural, unintended selection, then no organisms can have the power of real, intended selection. This move to exclude genuine agency was first made in the 19th century by Alfred Russel Wallace, leading him to disagree with Darwin's distinction between natural and artificial selection." Now, he projects this onto all modern evolutionary biologists. He cites an article by Jerry Coyne in the magazine Edge, decidedly not a scientific magazine, to make his case. In this article, Coyne is writing about free will and that he believes that it's an illusion. He writes that evolution has, quote, "bequeathed us with such a powerful illusion." Noble takes this as a critique of agency itself. If Coyne doesn't believe in free will, he must not believe in agency. And if he doesn't believe in agency, then organisms can't be choosing their mates intentionally. No free will, no agency, no intentionality. There's a lot wrong with this. I'm not especially interested in wading into the free will debate, but as we've already stated, if agency is simply to do something with a purpose, then organisms have agency virtually definitionally. Evolutionary biologists are hesitant about using words like agency because they have an anthropomorphic undertone and can be easily misunderstood by the general public. But it's not because we don't think organisms, including ourselves, make conscious decisions. But alas, Jerry Coyne speaks for us all, and apparently stating that free will is an illusion is akin to saying there's no such thing as intentional, i.e. sexual or artificial selection. Immediately after providing Coyne's quote, Noble writes, "That idea is itself an illusion generated by a conceptual mistake, which is to confuse the metaphor of selection with reality. If all evolutionary change is produced by natural, unintended selection, then no organism can have the power of real, intended selection." Nowhere in the article does Coyne state that only unintentional natural selection exists. Again, Noble's logic simply does not follow from Coyne's statement. It's also patently false. Entire research groups are dedicated to studying sexual selection. Indeed, evolutionary game theory treats organisms as decision-making, rational entities that not only make choices, but often make the best choices. Noble is either ignorant of this work or chooses to omit it. Noble goes on to argue that since consciousness itself is so metabolically expensive, it must have a purpose. He writes, quote, "Consciousness and the neural structures that support it and the capacity to make intentional choices are great consumers of energy. Their evolution required payback in terms of selective advantages, which I believe the nervous system has given us. Consciousness, like life itself, must be functional to justify its cost." No evolutionary biologist is saying that consciousness is free and has no function. Indeed, the function prescribed to the ability to make intentional decisions are the exact kind of functions that evolutionary biologists have long attributed to consciousness. In ecology, organisms are often modeled as optimal foragers, meaning that they are capable of discerning good from bad habitat and will make, generally, the most optimal decisions when it comes to how much time to spend looking for food in a given location. This obviously requires some degree of forethought, some intuition about sunk costs, some basic concept of purpose and agency. All these things are assumed in the most basic ecological foraging models and have been applied to everything from mammals to grasshoppers. And secondly, here and elsewhere, as we'll see, he's making an extremely adaptationist argument. He's assuming that everything that exists, every decision an organism makes, must be made for a purpose. Organisms don't do things for no reason. Indeed, applying too much agency can actually mislead. Organisms don't have the best information, so they can make bad decisions, or simply uninformed ones. And this exposes the danger of introducing words like agency and purpose. It implies organisms always do things for a reason. That is, that natural selection has shaped organisms to respond optimally in all situations. After all, they always have a goal in mind, right? Regardless, no evolutionary biologist would disagree that when organisms select mates, and by this I mean in organisms in which mate choice is actually a component of their reproductive strategy, they are generally doing so on purpose. That purpose may or may not be driven by a genetic predisposition to finding the best mate, but does sexual selection mean that organisms are directing their own evolution on purpose? No. This is a confusion of terms. Individuals don't evolve, and hence can't direct their own evolution. Only populations evolve, and so individual decisions on mate choice can cumulatively have an impact on the gene frequencies of populations. A single individual has no conception of this. They're not trying to change the population or influence future generations. Their purpose of action is simply to get a mate they like. This same confusion often occurs with respect to discussions of niche construction. Niche construction is another buzz phrase of both the EES and third way crowd. The basic idea is that organisms shape the environment that they are in. They construct their own niche. This can be done passively, such as when photosynthetic bacteria oxygenated the atmosphere, or actively, as when beavers construct dams and block creeks and rivers. By altering the environment, they shape how selection is expected to act, not only on the organisms doing the shaping, but also on all other organisms that happen to occupy that environment as well. So, by constructing their own niche, have organisms directed their own evolution? Again, definitions are important here. First, we should note, as pointed out by Richard Lewontin so long ago, that there doesn't exist an environment without an organism, or an organism without an environment. These two things are not finely demarcated, and they actively shape each other. Furthermore, organisms themselves are often considered part of the environment. We usually speak of the biotic versus the abiotic environment, but even these distinctions are squishy and feed back in on each other. If I'm in a forest, obviously trees are part of the environment, but so is the amount of sunlight that can reach the forest floor, which means forests are often cooler than grasslands. Temperature is a function of the abiotic environment, but it's being shaped by the fact that the trees, the biotic environment, are blocking sunlight. Both of these, temperature and trees, are a feature of the external environment of a bird that lives in the forest. Thus, it's once again a trivial statement to say that organisms shape the environment they're in. There is no environment without organisms, and organisms exist by literally interacting with the environment. These interactions, by necessity, alter the environment. Since the external environment can be a source of natural selection, altering it will alter the selective environment of future generations. There's nothing profound in any of this. Every ecologist knows these things to be true, as did Darwin and all of his contemporaries. Noble is, in effect, suggesting that a woodpecker creating a cavity in a tree is an act of purposive agency and directs the evolution of the nuthatch that might nest in it. Therefore, Neo-Darwinism is dead. The teleonomy of living systems is the result of two completely natural processes that are well understood within a modern synthetic framework. First is basic biochemistry. Cells respond in ways neither shaped by selection or impacted by genes because they are, after all, bags of molecules. And these molecules will chemically react in the presence of others, producing a myriad of responses. But there's no reason to provide purposive language to a lipid. Second, the ability to respond, shape, and interact with your external environment is a product of natural selection. organisms that failed to do so would be quickly weeded out. Words like intentionality, purpose, and agency, to me, really have no place in evolutionary biology, especially when evolution is being explained to the general public, because they have a tendency to confuse and give false impressions. This is nowhere clearer than when we see the Discovery Institute publishing articles about Denis Noble, claiming that agency and purposeness in organisms indicates that they were designed by an all-powerful God. Obviously, a bird foraging for seeds is not what they really think about when they think of agency, but that's the difference between how the public perceives these words and how they are being employed by Noble and friends. My point is that it simply isn't necessary to use such loaded words, as they indicate completely trivial concepts in the first place and only act to spread confusion about how evolution actually works. Now, there is a more direct way in which Noble, and in particular the biochemist James Shapiro, argue that organisms direct their own evolution. That is, they do so by directly modifying their own DNA. Shapiro calls this natural genetic engineering, a term that, as you might have guessed, has also been seized by creationists. But if it's true, it would certainly be a non-trivial action of purpose. Indeed, if an organism were capable of modifying its own DNA in service of adaptation, it would be a remarkable departure from the modern synthetic paradigm. While we mostly focused our critique on Denis Noble, a great deal of the third way rhetoric has been shaped by his accomplice, the University of Chicago biochemist James Shapiro. In the 1960s, Shapiro was at the forefront of work on the lac operon we've already discussed, working as a postdoc with both Francois Jacob and Jonathan Beckwith. In his work in Jacob's lab, he discovered that some mutations in E. coli were due to transposable element insertion. It's perhaps this early work in his career that led him to befriend Barbara McClintock, the discoverer of transposons. While most of his work has been focused on molecular biology, his interactions with McClintock inspired him to begin thinking about these ideas and how they relate to evolution. McClintock received the Nobel Prize in 1983 for her discovery of mobile genetic elements, which she first called controlling elements. She studied color patterns in the kernels of maize, noting that there appeared to be two elements that could move between chromosomes and, depending on their presence, alter corn color. Since these movements were cell-specific, it often led to variation in the color within a single ear of corn. Starting in the early 1990s, James Shapiro began to lay the groundwork for what would become his theory of natural genetic engineering, a centerpiece of which are transposable elements. As the work from McClintock and others show, mobile elements are capable of interacting with gene expression, as in the case of corn color, and thus as they move through the genome can cause an array of phenotypic effects. Shapiro, who firmly rejects the notion of junk DNA, arguing that all of the genome is useful and engineered by the organism, posits that transposons are under the control of the cell and are inserted purposefully around the genome as needed to regulate expression. He goes on to argue that things like the vertebrate immune system, which relies on generating genetic variation at the major histocompatibility complex, is an example of purposeful mutations that undermine the idea that mutations are random with respect to the need of the organism. Indeed, his book Evolution, A View from the 21st Century, published in 2011, is largely dedicated to debunking the notion that mutations are random and not guided by the organism for a specific purpose. He goes on to conclude in his book, quote, "Innovation, not selection, is the critical issue in evolutionary change. Without variation and novelty, selection has nothing to act upon." First, it has been long established that transposon mobilization is overwhelmingly harmful to the host genome, as would be expected if they originate as selfish genomic parasites. See my video, Your Genomic Parasites, to learn more about these curious features of the genome. Thus, in most organisms with large proportions of transposable elements, like ourselves, they are silenced. And if these silencers are removed, mobile elements wreak havoc on the genome. Thus, for natural genetic engineering to rely so heavily on transposon mobilization certainly casts doubt on how good of an engineer a given cell is. Next is that the evidence relies very strongly on these elements as responses to stressors. In a chapter Shapiro contributes to the book Evolution on Purpose, he includes a large table of when transposons are mobilized. All of them are either due to some kind of stress or due to hybridization. It has long been known that certain kinds of stressors are capable of removing methylation from the genome. Doing so would obviously have the effect of allowing transposons to mobilize as they are no longer silenced. Their mobilization is not evidence that they are responding to the stressor. Rather, the stressor has freed them from their prison. In the case of hybridization, transposon mobilization occurs because the hybrid offspring may not have the requisite gene for silencing a transposon. If one parent has the transposon, but the gene for silencing it is on a different chromosome, then there's a chance that the offspring receives the transposon one from one parent, but receives the chromosome lacking the gene for silencing it from the other. This leads to the transposon being expressed. As mentioned earlier, this is a case of hybrid inviability in Drosophila with respect to the P element. Now, Shapiro here might cry foul. He'd point out that there are ample studies documenting transposons contributing their regulatory apparatus to the host genome, and sometimes these can be helpful in stress response. But I contend this is a misinterpretation of the causal direction. To understand how this misinterpretation might occur, let's imagine a hypothetical organism living in a stable environment. This organism has selfish transposable elements that are presently repressed by methylation, preventing their spread. Suddenly, there's a massive heat wave, and this strips the methylation from the transposons. This allows them to start to proliferate and transpose into other parts of the genome. Transposons don't insert randomly. They can only insert in specific sites that have certain target motifs. Once it recognizes this site, it cleaves the double-stranded DNA, and with the help of an enzyme called reverse transcriptase, which it codes for itself, it can get inserted. Now, if there is a heat shock protein coding sequence that is downstream of the insertion site, this transposon insertion might actually increase the expression of that heat shock protein by recruiting transcription factors for itself. After all, transposable elements also recruit transcription factors. That's how they code for the genes necessary for transpositioning. The increased expression of the heat shock protein might prove beneficial. Our little hypothetical critter is able to survive the heat wave, where perhaps many others perished because they didn't get a transposon insertion near their heat shock proteins. Thus, we see here an element of randomness. That is, transposons insert randomly with respect to need, but not with respect to sequence. They don't know what the organism needs, they only know that their silencer has been removed, and so they can start doing their thing. Importantly, other members of the population may suffer from this, because the transposons may land in places that disrupt gene function, ultimately leading to cell death. But you wouldn't see the effects of these transposons because they don't survive for you to count. Hence, you're already biased to only observe those that landed in the right place to provide the benefit in the first place. This is a kind of survivor bias. Now, the big question, so often left unexplored in studies of shock on transposition patterns, is what next? Our hypothetical organism has survived the heat wave. Good for it. But now it has a transposon stuck upstream from a heat shock protein, causing excess expression even after the heat wave goes away. Even if the transposon gets methylated and silenced afterwards, the impact of this mutation is invisible after the event has occurred. Future descendants will inherit the mutation, but will not increase their fitness. The pressure for it to be there has already passed. So these sorts of punctuated stressors don't lead to sustained evolution, which is necessary for adaptation. Furthermore, it's often unclear from studies on transposition patterns that increased mobilization of transposons is actually helpful at all. Often they are merely correlating environmental stress with transposon activity. If stress removes methylation, as it is known to do, then all that is occurring are these genomic parasites are now free to proliferate once more. Indeed, the best evidence for this comes from the fact that most cancer cells show rapid proliferation of transposons, indicating a breakdown of the cell to contain these parasites. It's also important to note that these stressors, at least in multicellular organisms, almost always occur in somatic tissue, not in the germline. Thus, reorganization of the genome of somatic cells has no influence on evolutionary change. These reorganizations need to be passed on to offspring to be relevant. Now, none of this is to deny that transposons can and do get co-opted by the host genome to perform all sorts of tasks. But this co-option is done by standard population genetic processes. If a transposon gets inserted in a region upstream of, say, a nascent protein in need of a promoter, the protein can now make use of the transposon's promoter and be favored by selection. That is, in effect, a mutation. The transposon insertion followed by selection. Yet again, a classic Neo-Darwinian tale. Shapiro's natural genetic engineering idea clearly relies on the concept that most mutations that occur are directed by the organism, not random. However, if this were the case, it would lead to a fairly straightforward prediction. If we measure the distribution of fitness effects of de novo mutations, which include things like transposon insertions, they should be skewed towards being beneficial. After all, a natural genetic engineer surely wouldn't produce mostly deleterious mutations, right? Every distribution of fitness effects ever measured indicates that the vast majority of mutations are either deleterious or neutral, with very few being beneficial. Population geneticists Peter Kitely and Michael Lynch make this point forcefully in their 2007 article in Evolution titled "Towards a Realistic Model of Mutations Affecting Fitness," writing, quote, "The vast majority of mutations are deleterious. This is one of the most well-established principles of evolutionary genetics, supported by both molecular and quantitative genetic data." Furthermore, population genetic theory, going back to R.A. Fisher in 1930, predicts that the tail of beneficial mutations, if they arise randomly with respect to the need of the organism, should be exponentially distributed. This is because when a population is near an adaptive peak, most beneficial mutations should have very small effects. This has been the observed shape of virtually all beneficial mutation distributions. There's no reason to expect this distribution under a model of natural genetic engineering. Thus, if mutations are non-random with respect to the organism's need, and this is the result of natural genetic engineering, then organisms are engineering themselves, on average, to be worse off. Under Shapiro's logic, the evolutionary goal of the cell is to make itself less fit. I would be remiss in my duties if I didn't point out that one of Shapiro's skepticisms about the selection and mutation process to explain biodiversity is literally the waiting time problem, the same complaint that creationists make. In his book chapter contribution to the book Evolution on Purpose, he writes, quote, "It is very difficult to imagine how random mutations and phyletic gradualism could generate the functionally significant numbers of shared CRMs that appear at unlinked genetic loci encoding different proteins. The collective probability of the same sequence motifs independently evolving ab initio at multiple genomic locations is the product of their individual probabilities, and this product rapidly becomes impossibly small as the number of shared distal CRMs increases." This idea was debunked almost immediately after Michael Behe and David Snoke proposed it in 2004 in numerous papers. If you want to learn the details of why the waiting time problem is not a problem for standard evolutionary mechanisms, see my video, Waiting Time, No Problem. Shapiro and Noble also spend a lot of time complaining about what is known as the central dogma of molecular biology. While not necessarily a principle of evolution, it is one of the most grossly misrepresented ideas in biology, and so perhaps it's worth pointing out why they get it wrong. In fact, it's very likely you were taught the central dogma incorrectly. The central dogma is commonly stated as the pathway of DNA to RNA to protein. This version was popularized by James Watson in his 1965 book, The Molecular Biology of the Gene. But as Shapiro and Noble are quick to point out, proteins do modify DNA, and RNA can get transferred back into DNA. Since these protein modifications of DNA are, of course, an example of epigenetics, they insist that environmental induction of epigenetic modification is an example of the failure of the central dogma. But it's important to note that this version is not the original statement made first by Francis Crick in 1957. The original central dogma is, in Crick's words, quote, "the central dogma. This states that once information has passed into protein, it cannot get out again. In more detail, the transfer of information from nucleic acid to nucleic acid, or from nucleic acid to protein may be possible. But transfer from protein to protein, or from protein to nucleic acid, is impossible. Information here means the precise determination of sequence, either of bases in the nucleic acid or of amino acid residues in the protein." He restated it more succinctly in 1970 as, quote, "the central dogma of molecular biology deals with the detailed residue-by-residue transfer of sequential information. It states that such information cannot be transferred back from protein to either protein or nucleic acid." The original version of the central dogma is absolutely still true. The reason for this is the redundancy in the genetic code ensures that you cannot recreate the exact DNA sequence that coded for any protein, meaning that once DNA is translated into a protein, you can't back-translate it back to DNA or RNA because of the redundancy of the genetic code. Thus, Shapiro and Noble, when they complain about the central dogma, are doing so about Watson's version, which has been known to be false virtually since he stated it. Crick's version, the original version, remains true to this day. Lastly, Shapiro claims that innovation is the most important part of evolution because selection only sifts through innovations that it provided. In this, Shapiro and I are actually in agreement. Indeed, this was the argument of the mutationists like Hugo de Vries, Thomas Hunt Morgan, and Reginald Punnett. There are some who claim that selection is itself a sort of creative force of evolution, but I tend to disagree. Mutations are the creative force of evolution, but Shapiro avoids using that term, instead calling them innovations, because he wants to convey the impression that organisms are the cause of their own innovations. In reality, innovations are just mutations. They are changes in the heritable material that introduce something new to the population. The effect of that innovation is determined by its contribution to survival and reproduction, and hence its fate is dictated by the combined forces of drift and selection. And here again, we have a classic tale of Neo-Darwinism: mutation followed by drift and selection. Natural genetic engineering offers us nothing new. It's simply a rebranding of processes that have been understood since the 1960s. Shapiro's natural genetic engineering, it should be pointed out, is actually a kind of hyper-adaptationism. It assumes that not only is the entire genome functional, but that every mutation is directed by the organism. Thus, every gene duplication or transposon insertion must immediately have a purpose. Otherwise, it would indicate that there is unnecessary junk DNA, and there's no room for that in Shapiro's philosophy. As with Noble, we see that critics of Neo-Darwinism actually want something even more adaptationist. They want every feature of an organism to serve some purpose. This finally brings us to the last of Denis Noble and the third way's critique of Neo-Darwinism, his abhorrence of Richard Dawkins's notion of selfish genes. In this final section, we're going to finally find some common ground with Denis Noble. Some of his critiques here I quite agree with, despite the framing of them being extremely historically inaccurate. So in the next section, we're going to point out both where Noble gets things right and where he steers into left field. My hope by doing this one last is to try and show that I'm not just here to bash on these guys. Both Noble and Shapiro are highly respected in their fields. They're just not really evolutionary biologists. And it kind of shows, right? But that doesn't mean that they have nothing interesting to say, and in this next section, I think, is where we're finally going to see where they get some things right. Anyone who has read any of Noble's papers or listened to these podcasts he's been on know he's really, really obsessed with Richard Dawkins. Like, if he's going to cite a single evolutionary biologist, it's going to be Dawkins, and it's almost always his 1976 book, The Selfish Gene. I pointed out earlier that Noble thinks the fall of the Weismann barrier is one of the reasons that Neo-Darwinism is dead, and the other reason, to him, is the failure of what he calls the gene-centric view of biology. He credits this view almost exclusively to Dawkins, as if Dawkins not only came up with the idea of selfish genes, but that he has somehow risen to the level of an architect of the modern synthesis, that his ideas are akin to those of Ernst Mayr or R.A. Fisher or something, and all biologists have just accepted his word blindly as gospel. Like, here's a few clips of Noble going on and on about the selfish gene theory as if all biologists are on board, and Dawkins is somehow the new Darwin.
I do not think they have an answer. I debated all of this with Richard Dawkins in a broadcast which has gone viral in various parts of the world, and he had no answers to all the examples I gave. Again, I don't want to go into the technology, but one example after another that I gave to him, he diverted off to use his usual mantra about genes are causes of what we are, rather than address the detailed examples I gave. Everybody can look that up. Everybody can find all the literature that I cited too. It's all there on the website. A year and a half ago, I debated the whole question with the main exponent of Neo-Darwinism, Richard Dawkins. Both he and I, we've been talking with each other, and we have a sort of friendship between us for over, well, nearly 60 years, because I examined his thesis in 1966. The selfish gene came out 10 years later. Now, we both respect each other, but he was unable even to understand the molecular, biological, and cellular points and explanations that I was giving.
Neo-Darwinism that became the selfish gene, aka the modern synthesis or gene-centered theory of evolution, is that all the same thing?
That's it. That's the best definition. You've just given it, yes.
The fact that he equates the modern synthesis, Neo-Darwinism, and gene-centrism as the same thing is a stunning testament to his misreading of the history of evolutionary theory. Like, to virtually all of the architects of the modern synthesis, as well as Fisher, Wright, and Haldane, natural selection acted on individuals, not on genes. That is, the modern synthesis is not gene-centric. It's easy to get confused into thinking it is. Indeed, most people think that population genetics is gene-centric because it models changes in gene frequencies. But this is incorrect. Fisher, Wright, and Haldane considered selection's effects on genotypes, not on alleles. A genotype represents the phenotypic expression within individuals. For example, let's say there are two alleles, A and B, and A is dominant to B. These alleles code for flower color. If you have the A, you're a red flower, while B is a white flower. Thus, individuals that are AA and AB are red, and those who are BB are white. Now, if I said selection is favoring red flowers, I could model this at the genotypic level by calculating the fitness of each genotype. If P is the frequency of the A allele and Q the frequency of the B allele, then the mean fitness of the population is W bar equals P squared W sub AA plus 2pq W sub AB plus Q squared W sub BB, where the W's with the subscripts represent the fitness of each genotype. Notice that despite the fact we are modeling genes, the fitness is about the phenotype. Are you red or white? And hence selection is acting on the individual. Selection on individuals has the effect of changing allele frequencies, since these alleles are what ultimately give rise to the phenotype. But that doesn't mean that population geneticists believed that selection was acting directly on alleles. In fact, Ernst Mayr was a vocal opponent of the idea that selection acted directly on genes. Indeed, he opposed reductionism in all of his writings, arguing that selection shaped the entire organism, not favoring atomized parts. Gene-centrism emerged after the modern synthesis, starting with George C. Williams in his 1966 book Adaptation and Natural Selection, but also John Maynard Smith and W.D. Hamilton, who often modeled genes as independent units from their carriers. These ideas were popularized by Richard Dawkins in his 1976 book The Selfish Gene. But notice how these ideas were not part of the modern synthesis and were vehemently attacked by the synthesizers, including Ernst Mayr, but also Sewell Wright. So it isn't necessary that we accept that genes are the unit of selection to be on board with modern evolutionary theory or the modern synthesis. But Noble's critique is actually a bit more subtle than this. In his interview with Andrea Morris, he says the following, quote, "Where are all the genetic cures? They don't exist. Where will they be? They won't exist." He was referring here to the Human Genome Project, a multi-billion-dollar project that promised to deliver the answers to so many human ailments. If only we could identify the underlying genetics of things like heart disease, diabetes, and cancer, we could find cures for them. But to people like Noble, and myself for the record, this promise was built on a false premise. It was assumed that all of these things even have a genetic basis in the first place. However, in the 20 years since, we still don't know anything more about the genetic basis of any of these big killers. It turns out there simply doesn't appear to be any clear one-to-one relationship between genes and complex disease. To Noble, this proves gene reductionism, which to him is the logical consequence of seeing biological entities as merely vehicles for their selfish genes, is utterly false. I think he slips a bit here. The goal of the Human Genome Project, after all, is to save the vehicle of the genes by understanding how the genes construct the vehicle. It's not necessarily the case that all these scientists thought that vehicles are unimportant or that genes are all that matter. Noble often does this. He stretches Dawkins' analogy farther than I think Dawkins intended. He frequently cites the famous quote from The Selfish Gene that genes created us body and mind, which sounds rather damning, but there is more nuance that Dawkins inserts than what Noble portrays. For example, Dawkins clarifies later in the chapter that what he means is that genes are the necessary prerequisite for a body and mind to be built, but that doesn't mean that they dictate what the body and mind become. He gives an example of a computer that plays chess, with the person coding the algorithm for the computer representing the genes, and the computer the vehicle. The person writes up the algorithm that the computer draws from, but it doesn't include all possible moves of the chess game. The number of possible chess games is astronomically high, around 10 to the 120th power, greater than the number of atoms in the universe. Instead, the programmer gives the basic rules and goals of the computer, but then the computer plays the game. The underlying algorithm is flexible enough that the computer can respond dynamically to the game at hand. In this way, genes provide the basic rules to the vehicles, but it's ultimately the vehicles that have to interpret the world around them. This is a bit different than what Noble means when he says genes created as body and mind. Now, Noble is correct about the hype around the Human Genome Project and its utter failure to deliver on its promise. I think the lesson here is that complex diseases have a substantial environmental component, and while there may be some underlying genetic variants contributing to them, changing the environment is a much better route to medical intervention. For example, even if someone has many genetic variants that predispose them to having heart disease, creating a pill that serves to counteract these variants does nothing if a person eats a cheeseburger every day of their life. Instead, we'd have much more success ensuring people have access to quality, healthy foods at affordable prices and have education to understand the importance of their dietary choices on their health. I'm also sympathetic with Noble's critique of genetic reductionism writ large because it can slip dangerously close to hereditarianism, the belief that all human differences are the result of genetic differences. See my video, You Are Not Your Genes, for an exploration of this concept. Ultimately, whether or not genes are the target of selection, which was ultimately Dawkins's argument, is irrelevant to the current standing of evolutionary theory. Quantitative models of evolution assume that selection works on the cumulative effects of genes and the environment, not merely on individual selfish genes, so it's unclear to me if Noble is just strawmanning evolutionary theory or is simply unfamiliar with the technical details of it. Honestly, this has been the consistent theme throughout. The third way supporters have consistently misrepresented or just misunderstood the history of the development of evolutionary theory. They've strawmanned arguments by the proponents of the modern synthesis and downright ignored existing theoretical models that readily accommodate all the things that they say are missing. The third way of evolution was launched in 2014 by Denis Noble and James Shapiro. In 2016, they held a much publicized meeting at the Royal Society, organized by Denis Noble and other supporters of the EES, including Kevin Lala and John Dupree. The meeting kicked off a media frenzy over the current status of evolutionary theory. Everyone was excited that finally there was some controversy in a field that had felt settled for so long. David Shuker, an evolutionary biologist at St. Andrews, was in attendance at the 2016 meeting. I want to read you an account of his interaction with Denis Noble as told by the science journalist Carl Zimmer. After listening quietly for a day and a half, the University of St. Andrews researcher had had enough. At the end of a talk, he shot up his hand. The talk had been given by Denis Noble, a physiologist with a mop of white hair and a blue blazer. Noble, who has spent most of his career at Oxford, said he started out as a traditional biologist, seeing genes as the ultimate cause of everything in the body. But in recent years, he had switched his thinking. He spoke of the genome not as a blueprint for life, but as a sensitive organ, detecting stress and rearranging itself to cope with challenges. "I've been on a long journey to this view," Noble said. To illustrate this new view, Noble discussed an assortment of recent experiments. One of them was published last year by a team at the University of Reading. They did an experiment on bacteria that swim by spinning their long tails. First, the scientists cut a gene out of the bacteria's DNA that's essential for building tails. The researchers then dropped these tailless bacteria into a petri dish with a meager supply of food. Before long, the bacteria ate all the food in their immediate surroundings. If they couldn't move, they died. In less than 4 days in these dire conditions, the bacteria were swimming again. On close inspection, the team found that they were growing new tails. This strategy is to produce rapid evolutionary genome change in response to the unfavorable environment, Noble declared to the audience. It's a self-maintaining system that enables a particular characteristic to occur independent of the DNA. That didn't sound right to Shuker, and he was determined to challenge Noble after the applause died down. "Could you comment at all on the mechanism underlying that discovery?" Shuker asked. Noble stammered in reply. "The mechanism in general terms, I can, yes," he said, and then started talking about networks and regulation and a desperate search for a solution to a crisis. "You'd have to go back to the original paper," he then said. While Noble was struggling to respond, Shuker went back to the paper on an iPad, and now he read the abstract in a booming voice. "Our results demonstrate that natural selection can rapidly rewire regulatory networks," Shuker said. He put down the iPad. "So, it's a perfect, beautiful example of rapid Neo-Darwinian evolution," he declared. This interaction perfectly encapsulates the debate. The EES and third way crowd are lost in the weeds. To quote R.A. Fisher once more, they are buried under masses of detail that only act to obscure principles. Noble was so focused on the neat specifics of the biochemistry of the experiment that he missed what was staring him in the face: an excellent study on the power of natural selection. On closer inspection, this is the fate of all third-way claims. They are specific details about specific systems. They are the proximate mechanisms, but what evolutionary theory is all about are the ultimate mechanisms. What is the source of the origin of these systems, and what allowed them to spread and be maintained in a population? The answer to these questions lie in the basic modern synthetic paradigm: mutation, selection, and drift. Perhaps the most frustrating thing I experienced while researching for this video is the lazy dismissal of evolutionary biologists by people like Denis Noble. When asked why it is that evolutionary biologists haven't accounted for their mechanisms, if indeed they are so obvious, Noble brushes it aside. In the Forbes article, Morris writes, quote, "Noble attributes our legacy of missteps to rigid assumptions put in place over a century ago to stand in for a lack of evidence." Rigid assumptions? That's it? Evolutionary biology was hindered by rigid assumptions. This brazen disrespect for the history of the field is insulting. As I pointed out in the historical section at the beginning of the video, evolutionary biologists came to the current paradigm not by rigid assumptions, but by evidence. We conducted experiments. We constructed mathematical models. We examined 100 years of data on crop and livestock breeding. All of these things and more contributed to why the modern synthesis happened in the first place, and why its basic principles still underlie evolutionary theory today. But two can play this game. The insistence on words like agency and purpose, not to mention the deep hatred of reductionism that runs through third-way writing, makes one suspicious that there are also ideological reasons for rejecting Neo-Darwinism and really population genetics as a whole. Sean Carroll, in his book Endless Forms Most Beautiful, states that population genetics is uninspiring, that we should be telling students about zebras and butterflies instead of alleles and gene frequencies. Mike Lynch said it best in 2007 when he wrote in response, "Evolutionary biology is not a storytelling exercise, and the goal of population genetics is not to be inspiring, but to be explanatory." The historian of science Peter Bowler, in his 1989 book Evolution: The History of an Idea, argued that the consistent attempt to revive Lamarck was because, quote, "Lamarckism allows life itself to be seen as purposeful and creative. Living things are in charge of their own evolution. They choose their response to each environmental challenge and thus direct evolution by their own efforts. With or without any religious implications, this is certainly a more hopeful vision than that derived from Darwinism. Life becomes an active force in nature, not merely responding in a passive manner to environmental pressures." Indeed, recently Denis Noble appeared on a Christian podcast alongside a creationist in which they argued that life was clearly purposeful. While Noble didn't make any religious claims himself, he did not correct the creationist when he said that living things clearly have higher purpose, conflating teleonomy with teleology. Furthermore, during his time as editor-in-chief at the journal Progress in Biophysics and Molecular Biology, he oversaw the publication of 4 papers by a pair of creationists, Olin Brown and David Hollander. Thus, whether or not Noble himself holds pseudoscientific ideas seems a bit irrelevant. He's willing to share a platform with them, which is damning enough. Shapiro has also catered to creationist and intelligent design language. In his chapter in Evolution on Purpose, he critiques Neo-Darwinism as a physicalist reductionist theory. While physicalism can technically be distinct from materialism, it's hard to imagine what kind of theory he thinks is replacing it. A non-physical one? What's a non-physical theory in biology? We're left with the impression that he thinks terms like agency and purpose are themselves driving forces that are not merely emergent properties of chemicals, but are something special. This kind of language walks dangerously close to vitalism, spiritualism, and frankly creationism. Again, there's a reason creationists latch onto the language employed by the third way. If you consider yourself a respectable scientist, but when you look around at all of your supporters and you find yourself in a sea of conspiracy theorists, it might be time to reevaluate what kind of message you're sending. So there you have it, folks. 10 years after the third way was first proposed, it still remains a fringe idea in evolutionary biology, with most evolutionary biologists having no idea what it is. This is because evolutionary biology, despite what the popular media would have you believe, is an incredibly robust and mature discipline. As I have done on this channel many times before, I urge caution to all of you that consume popular media and press releases and interviews and whatnot on evolution. If you see a headline like, "We need to rewrite the textbooks," or "Evolution may be purposeful and it's freaking scientists out," 99% of the time, it's clickbait. The reason Noble has become so popular on YouTube is not because he's an important evolutionary scientist. It's because he's feeding the algorithm what it likes: controversy. No one likes to hear, "It's actually all good here." When Brian Charlesworth said that most of the major issues in evolutionary theory had already been solved, Esser Piglucci lamented, quote, "Well, perhaps, but some of us are not ready for retirement just yet." One of the greatest neo-Darwinists of our time, Joe Felsenstein, regarded as the father of phylogenetics and the phylogenetic comparative method, once wrote an excellent review of the 1986 book Evolutionary Theory: Paths into the Future. In the book, the various authors tried to argue, as Denis Noble is now, that Neo-Darwinism is dead. By the end, Felsenstein remained unconvinced, writing, quote, "We are left at the end still waiting for a post-Neo-Darwinian theory that has not appeared." Almost 40 years later, and after many new iterations on the same old critiques, we're all still waiting for a better theory that has yet to materialize. Thanks everyone for being here. If you enjoyed this video, hit like and subscribe, and drop a comment if you have any questions, thoughts, or concerns. If you'd like to support the channel monetarily, I've included an Amazon wish list in the description below. Take care, and I'll catch you next time.
It's a kind of community of people who defend the fortress, if I might put it that way, of Neo-Darwinism. I've never, in my long career in science, encountered anything like it. I've had controversies in my own field of physiological science, but I've never encountered a situation where there's a kind of dogmatism in reverse.